Another important point to discuss in this section is sterilization techniques, because we have said that maintenance of clean is mandatory. Contamination in plant tissue culture consists of impurities, chemicals, or pathogens. These make the cultured tissues unfit for further development of healthy plantlets. It is also difficult to get rid of contamination completely as plants are highly susceptible to pathogen infections. There are different contaminations that can occur in your tissue culture experiment. Microorganisms could be present on beakers, pipettes, or on your culture vessels. A single fungal spore or bacterial cell that comes into contact with the culture media or explants rapidly contaminates plant tissue culture materials and instruments as well. The tissue culture media also contains sugar and other elements that are more susceptible to attracting microbes like bacteria, fungi, etc. Thus, plant tissue cultures are naturally prone to contamination and hence are hard to avoid. Over the years, scientists have come up with different solutions to reduce existing contaminants. But contaminations are still one of the largest bottlenecks in the world of plant tissue culture, leading to loss of yield and productivity among cultured plants. We will group sterilization techniques into three major categories. 1. Sterilization of media and small instruments. 2. Maintenance of aseptic condition. 3. Explant sterilization. Let us now discuss different methods of sterilization that are popularly used for eradicating or avoiding contaminations. Sterilization of media and glassware. Many laboratories use autoclaves for sterilizing media, which works on the principle of moist heat. Moist heat sterilization is a procedure in which heated, high-pressure steam is used to sterilize an object at 121 degrees centigrade, 105 kilopascal pressure for 15 minutes. You can sterilize labware, glass articles, pipettes, and culture media with the help of an autoclave. However, using autoclave as sterilization technique for media sterilization has both advantages and drawbacks. The advantages are speed, simplicity, and total destruction of viruses. There are also some important considerations you should give when you are using autoclave for media sterilization. These are: the pH of the media is lowered by 0.3 to 0.5 units. Autoclaving at too high temperature can caramelize sugars, which may be toxic. Volatile substances can be destroyed by the use of an autoclave. Autoclaving for too long periods can precipitate salts and depolymerize the agar. Thus, care should be taken to use the correct duration and temperature. 2. Maintenance of aseptic condition. Generally, this sterilization protocol is done in a transfer room, and we have a combination of different sterilization protocols to eliminate microbes. One of the main ways is using air sterilization. In simple terms, there are millions of particles floating in the ambient air. They may comprise microbes such as viruses, bacteria, fungi, and many more. Eventually, these floating particles will land on objects such as culture vessels, explants, etc., and cause contamination. For this very reason, it is necessary for you to ensure that clean and sterile air is flowing over your tools, containers, and specimens. In order to achieve this goal, you need proper air treatment equipment such as laminar flow cabinets. They possess HEPA filters, high-efficiency particulate air filter, and hence, the air passes through this filter and thereby traps fungi, bacteria, and other dust particles. You also need to sterilize the surface areas in your laboratory. This means that all the surfaces such as tabletops, floor, cabinets, cupboards, etc. should be sterilized with ethanol, bleach, or a cleaning solution on a regular basis. Explant sterilization. Explant sterilization is a crucial step in the tissue culture process. The explants that you obtain from the stock plant need to be free from any kind of microorganisms. This method involves several steps of treatment for efficient results. Initially, you need to clean your explants after extracting them from the stock plants. You can decide on which type of cleaning protocol to use based on how contaminated your explant is. You also need to consider the sensitivity of the chosen explant to the cleaning agents/disinfectants. A too strong cleaning agent will kill your explant, while a too weak agent will not do the job. Let us now talk about some common sterilizing agents. Common explant sterilization agents. 1. Calcium hypochlorite. It is commercially available in the form of powder, and before using it for sterilization, it must be dissolved in water. You also need to filter this solution. It is normally used in a concentration of 3.25%. 2. Sodium hypochlorite. You can also call it bleach. It is one of the most commonly used agents for surface sterilization of explants. Bleach is diluted up to 10 to 20% for sterilization in tissue culture processes. This gives you a solution with the final concentration of 0.5 to 1% of bleach. The time duration required to sterilize the explant varies for each type of explant, but usually 30 to 40 minutes are enough to serve the purpose of sterilization. 3. Mercuric chloride. It is rarely used in labs because of its high toxicity to plants as well as humans. So, if you're using it, follow extensive care. 4. Hydrogen peroxide. It is also a commonly used chemical for surface sterilization. The most favorable concentration of this agent is 3%. However, you should take all precautions while using this agent for sterilization. 5. Ethanol. It is also known as isopropyl alcohol. The ethanol concentration between the range of 70 to 95% is extensively used for sterilizing. However, ethanol is also toxic to plants. Hence, you should keep explants in ethanol for only 1 second as a longer duration in ethanol damages the explants.